Thanks, Kat, and good morning and welcome to everyone. So as Kat said, my name is Rob Signer. I'm an assistant professor in the Division of Regenerative Medicine, and I'll be chairing this morning's first session, the topic of which is regenerative medicine. Now, if any of you have had a chance to look at the schedule so far, you might be surprised to see that the three talks under the regenerative medicine category actually all have to do with cancer and not regenerative medicine per se. This is actually pretty poignant, I think, because cancer is really the evil twin of regenerative medicine. You see, regeneration in our bodies is really driven by rare populations of stem cells that reside in most of our adult tissues. And those stem cells really have this incredible capacity to self-renew throughout our lifetime and to differentiate and regenerate all of the different mature cell types within our tissues. And what's really incredible about these stem cells is that they can respond to all types of different cues and turn on and turn off with extreme precision. And it's the loss of that precision that can really lead to the development of cancer. And this can happen in a couple of different ways. It can happen when stem cell mechanisms become, overactivated and lose control, or when they become co-opted or hijacked by the wrong types of cells that really lead to out-of-control growth in cancer. And so to really, illustrate this point, I always tell people, stem cell biology is cancer biology. And by understanding really how stem cells work gives us a better insight into what goes wrong in the context of cancer and how to better target and treat cancer cells. And on the flip side, understanding the basis of cancer helps us to identify new genes and pathways that are really instrumental for harnessing the regenerative potential of stem cells and treating a wide array of human diseases. Now, one of the absolute pioneers that has led our field in understanding this connection between normal stem cells and cancer cells is our keynote speaker for this morning, Dr. Jane Visvader. So Dr. Visvader earned her PhD in molecular biology in the lab of Robert Simons at the University of Adelaide. And then she went on to do postdoctoral work, actually right next door at the Salk Institute with Inder Verma, and then back in Australia in Jerry Adams' lab, where she actually discovered important regulators of hematopoiesis. She carried on that work as an instructor at Harvard Medical School, working alongside Stu Orkin. And then there was a sort of change in direction in 1998. She was recruited to the Walter and Eliza Hall Institute for Medical Research in Australia, where she's now a professor and joint head of the Division of Stem Cells and Cancer. So Dr. Visvader has made a number of really seminal discoveries in the fields of mammary stem cells and breast cancer. Her 2006 Nature paper describing the prospective identification, isolation, and characterization of mammary stem cells really remains one of my favorite papers in the entire stem cell field. It's a really elegant paper. And if there are students or postdocs in the room that consider themselves serious about stem cells and haven't read this paper, you should go home at the end of the day and read that paper. This was at a time when most people couldn't even figure out how to sort a live epithelial cell. Dr. Visvader was, sorting and isolating these rare populations of mammary stem cells and doing single cell transplantations to regenerate entire functional mammary glands. And so, this paper really sort of launched what's been now over a decade of a series of really high impact and high profile papers that have gone on to characterize the cell intrinsic and extrinsic regulators of mammary stem cells. She's identified how developmental changes can alter the plasticity or differentiation potential of these stem cells, and most recently has actually described that there are multiple different types of populations of mammary stem cells. And in parallel with all this, she's taken those discoveries and helped us to understand the cell of origin of breast cancer and which stem and progenitor cells breast cancer is really coming from. Dr. Visvader has received a number of incredible awards, including the GlaxoSmithKline Award for Research Excellence in 2008, the Royal Society of Victoria Research Medal for Scientific Excellence in 2014, and the Lemberg Medal in 2016. Dr. Visvader, we're thrilled that you're here. We look forward to your talk, which is titled Mapping the Epithelial Hierarchy to Understand the Origins of Breast Cancer.